So we are going to talk about now relationship between genetics and epigenetics and the origin of word epigenetics and why we call this epigenetics. So what we have just seen, we learned about relationship between genes and chromosomes, and actually what is a gene. That is what we just covered. What we are going to learn now, the genetics and epigenetics. We have these chromosomes, existing in a eukaryotic cell like this, and these chromosomes, they get inherited through mitosis. You should watch videos about mitosis and cell division. We talked in great detail there. So after cell division, these chromosomes, so let's say we are talking about mitosis. So after mitotic cell division, the mother cell, the division of mother cell, it leads to generation of two daughter cells which are identical to mother cell in terms of the genome, the chromosome numbers. Now, since genes, they reside on chromosomes, from mother to daughter cells, this is mitosis, for example. And if you go to the meiosis, so I just try to, so both the pairs of chromosomes, the homologs. So in mitosis, these chromosomes are inherited equally, so the two daughter cells, they look exactly identical to the mother cell in terms of number of chromosomes. In case of meiosis, what we said, if we consider the same cell, we know that after meiosis we have, so this is the diploid state, we will have haploid cells, and each of the haploid will inherit like this. Half the number of chromosomes as compared to the mother cell. Essentially what I'm trying to tell you, that genes through inheritance, either in mitosis or meiosis, are inherited to future generations, and this whole thing is called genetics, because I want to eventually explain you the epigenetics so that we move on to the concept of epigenetics in development or epigenetic cell memory. When you are inheriting either through mitosis or meiosis, basically you are going through, this cell cycle. Say you have G1, S, G2, and then you have the mitosis or meiosis. Here in the S phase, you replicate, DNA replication takes place, and all those chromosomes, metaphase chromosomes, where each of this chromosome will have sister chromatids, and then through prophase, metaphase, anaphase, etc., the inheritance takes place. This inheritance of chromosomes, either during mitosis or meiosis, that makes the crux of genetics. Genetics is all about genes and inheritance of genes to daughter cells or to next generation through meiosis. Okay. Now, if there are any errors, any mutations in a gene, remember our genes, protein coding gene or non-coding genes. For this particular section, we will focus on protein coding genes so that the concept of learning epigenetics becomes easier and simpler. So, if there is, we assume let's assume this is a protein coding gene. Just write down its so this is the stop codon, this is the start codon. Imagine you have, this is the wild type sequence of a gene. And if there is a mutation in this gene, same DNA sequence, same gene, it's the same gene. Imagine there is a mutation here at this particular point. So let's write down the sequence. So here, this sequence is GAC. This is also GAC. Imagine due to this mutation, this G is converted into T. This is going to result in change in an amino acid. So GAC, let's say it encodes an amino acid B, and due to this change of G to T, it is going to result in change in an amino acid, a missense mutation, and it's now an amino acid X. Now, this is the wild type, this is the mutant. This gene present here in chromosomal context, when DNA replication will take place, when here either through mitosis or meiosis, inheritance of this gene will take place. Here you will have a variant, a mutant protein. This is the wild type. And generation after generation, this mutation will inherit like this. This is the fundamental genetic principle. Okay? However, now let's look at, so this is what we just said. This is Genetics 101. This is fundamental genetic principle. Once you have a point mutation due to whatever reason, this mutation in a mutant will inherit through mitosis, through meiosis, etc. So if it is in a germ cell, it will inherit through meiosis. If it is in the somatic cell, it will not go through meiosis to the next generation. This is fundamental genetics by the Bio 101 we learned. Now, this was genetics. Now let's look at the epigenetics. The same gene, okay? So, of course, mutation cause disruption of gene function, or suboptimal function of gene, or loss of gene function altogether. So, which means in case of wild type, if this protein is performing a function, in the mutant it's either performing suboptimal function or it has lost altogether its function. In case of epigenetics, the same gene Now, look at the wild type sequence as well as sequence in the potential mutant. Now, there is no mutation, there is no change. The sequence between wild type ATG, ATG, CAG, CAG, CGA, CGA, GAC, GAC, CAT, CAT, and stop codon. And there's always promoter sequence, etc. as well. Now, there's no change in the DNA sequence. Yet, you may lose function of the gene. Here, gene is normally functional, getting transcribed, translated, protein is getting made, and protein is performing function, which means gene is functional. Here, you lose gene function, although all the sequence is same. What has happened? What we have learned that DNA, it may get modified. DNA or these histone proteins. So the fun part starts now. The fascination starts now. These nucleosomal proteins, H2A, H2B, H3, H4, or the DNA, the two components of the chromatin, DNA or histone octamer, and histone octamer is H2A, H2B, H3, H4, either of them, they become chemically modified. DNA sequence remains same. No change in the DNA sequence. Whether it's the promoter sequence, it's gene region, the gene body, it remains same, but due to these chemical modifications. For example, in case of DNA, you have, cytosine in CG contact, it gets methylated. So we call this 5-methyl cytosine. And other than this, the histone octamer, which was this H2A, H2B, H3, H4, these proteins, amino acids in these proteins, they get covalent modifications or chemical modifications. What is the consequence of these modifications? The consequence is, if you have DNA methylation, I just told you 5-methylcytosine. We will call this symmetrical methylation because it's a symmetrical sequence, like a palindrome, CG and then in the second strand CG. Whenever you will have 5-methylcytosine in the promoter elements, the consequence is gene silencing. RNA-Pol2 machinery cannot transcribe that gene and you have loss of gene function. No messenger RNA is being synthesized. The histone octamers, let's say I draw here H3. This is the protein. This is its N-terminus. So proteins come in amino and carboxyl terminal. Like DNA has 5 to 3 orientations, proteins have amino and carboxyl terminals. Now, the N-terminal tails, these vertical lines I am drawing, they represent different amino acids. And these amino acids, they can be chemically modified. For example, methylation of lysines or phosphorylation of serines or methylation of arginines. So these covalent chemical modifications of different amino acids on histone proteins, they can also have consequences on the gene activity. So without changing the DNA sequence of the gene, you have resulted in loss of gene function. The gene has lost its function as compared to wild type. Why we call, and this is called epigenetics. Epi means above genetics. So the basic genetic information, the basic gene code remains same, no change, but above the DNA sequence, you have these modifications or changes either on the DNA or histone proteins, which result in loss of gene activity or gene function because the transcription is being blocked, or messenger RNA. There are also times when this gets transcribed but messenger RNA gets degraded. So we call this epigenetics because these codes, which are histone modifications or DNA methylation, whatever, they get inherited through mitosis. In certain cases, they inherit through meiosis as well. Which means if you have such modification of the gene sequence in a nucleosomal context, which means DNA methylation or histone modifications, they will inherit during mitosis or meiosis. That's why the genetics part comes in. The inheritance. Inheritance of coding information makes the crux of genetics. So epigenetics information also is passed through mitosis. So you have DNA methylation. It passes through mitosis, and gene activity, which was silent, remains silent. We call them, so genetic mutant is this one. These ones are called epimutants. They are not classical genetic mutants. They contain, chemical modifications of the genome, which does not change any primary sequences of the gene, but in terms of gene activity, in terms of their protein function, we have lost them in epimutants as well. Good thing is epigenetics is reversible. For example, let's now look at just one example I give you, the DNA methylation. So, for example, here, this is this gene sequence. You have CG, CG, let's say in the promoter sequence, and it gets methylated. We call this symmetrical because it's a symmetrical sequence. There are asymmetrical DNA methylation as well. We will come across them later on. When it gets replicated, when DNA replication takes place, the mother strands, okay, so the CG, CG, and here the parental methylation. Now the second strand, the new strand of DNA is synthesized, and this half methylated or hemimethylated state during DNA replication, during the S phase, is recognized by DNA methyltransferases, the enzymes which catalyze methylation. And one of the enzymes which is called DNA maintenance methyltransferase, in the newly replicated strand, it will add, DNA methylation. So the epigenetic code is inherited through DNA replication. If it is mitosis, the two daughter cells, they are going to inherit the same structure and function, chromatin structure or function. So if gene was off here, if the methylation is in the promoter region of the gene is being inherited in the off state in the daughter cells as well. So that's why we call this epigenetics, because you have an initial stimulus which resulted in chemical or covalent modification of DNA or chromatin, and that if that in the absence of stimulus, that information is inherited in the form of loss of gene activity, we call this epigenetics, because the basic DNA sequence, it remains the same. There's no change in the DNA sequence. It's reversible because these modifications, they can be reversed as well. There are histone, if there are histone phosphorylation is taking place, histone dephosphatase are there. Histone phosphatases are there. If methylation is there, there are histone demethylases as well. Okay? Or if there is, let's say, ubiquitination is there, deubiquitinases are there as well. So now I hope the difference between genetics and epigenetics is clear. In case of epigenetics, it's basically study of changes in the gene expression caused by certain molecules or signals which do not alter the basic DNA sequence, but they result in changes in the gene expression which are heritable through mitosis and in certain cases meiosis, for example, imprinted genes in humans or in mammals. So now, once we have understood what is relationship between genes and chromosomes, what is a gene, and what is genetics versus epigenetics, in next part we are going to cover in greater detail the different components, different mechanisms of epigenetics which result in such kind of modifications, and consequent loss of gene function or gain of gene function can also be. It can be in the one cell, for example, gene is off, in the other gene is on. For example, a gene is on in the eye cell, but same gene is off in the heart cell. That is due to epigenetics. So in the next section, we are going to talk about epigenetic cell memory now, and how actually epigenetic cell memory contributes to cell fate determination and inheritance of cell fate, which is established very early during development. So stay tuned.